Okay, move on. Now the process of gene cloning is used to locate the copy a specific gene from all the DNA in an organism. This bacteria contains a single gene of interest, which is a red color here in the red segment here. The genetic engineers want to clone this part and insert it into another organism. Okay. The DNA is first extracted from the bacteria into the test tube using extraction techniques. A restriction enzyme is added to cut the extracted DNA into the gene-sized species. In another test tube, extracted bacterial plasmid are cut using the same restriction enzyme, because restriction enzyme will cut a particular location and that particular location is specific to attach with another segment of gene. The cut plasmids are then mixed together with the gene-sized species of the DNA. The gene-sized pieces join and bond with each other to cut plasmid to form recombinant plasmids. Some of the cut plasmids may bond with themselves without a piece of the cut extracted DNA. Bacteria are then added to the test tubes of the recombinant plasmid. Electroporation uses quick pulses to the electricity to create small holes in the bacterial cell. Recombinant plasmids are then able to enter the bacterial which will express the genes. The transformed bacteria will be considered transgenic. However, not every bacterium will eventually take all those plasmids but become transgenic. Plasmids naturally contain an antibiotic resistance genes, therefore transformed bacteria will be antibiotic resistant. After electroporation, the bacteria are plated out onto the medium containing a particular antibiotic against which the resistance is developed for those transgenic bacteria. Bacteria that have not been transformed will not have the antibiotic resistance genes and they will die. Now bacteria that have been transformed with the plasmid will grow and multiply into the colonies that are visible to the naked eye. Organisms have thousands of genes. It may require hundreds of petri plates of bacteria to clone all of these genes. This collection is called a gene library. The genetic engineering must then find which bacteria colony has been transformed with the gene of interest. That colony is then transferred to the sample petri plate. It is allowed to multiply and divide into large colony with hundreds of copies of the gene of interest, and we will find our gene of interest inserted into the desired bacterial cell or desired individual cell. That's how we can clone a gene onto a desired individual.